Okay, so today we want to talk about oncogenic viruses, and we know we have viruses that can cause tumors, especially in case we have immunocompression. Okay, so this will bear the topic viral oncogenesis, and I want us to talk about it right now very quickly. So we have the first that we want to cover is called the HTLV1, and this one will cause the adult T-cell leukemia or lymphoma. The adult T-cell leukemia or lymphoma. A tumor this tumor is basically endemic in some parts of Japan, Caribbean Basin, South America, and even some parts of Africa. And in some countries, wherever you want to do a blood transfusion, it is usually mandatory that you do a test for this particular HTLV1. That's why you'll find that especially in countries like Japan, even the Caribbean Basin, just to mention a few, you'll find that we usually carry out a blood test of this particular virus because it is endemic in these regions, okay? So similar to HIV, it has tropism for the CD4 T cells. Remember, we have two types of tropism for HIV. We have the dual tropism, we have the R5, and we have the X4 tropism. But this dual tropism, especially for the CD4 positive cells, are shared both by HIV virus and the HTLV1 virus, okay? Hence, this subset of T cells is a major target for neoplastic transformation. So because it has a tropism for CD4 T cells, this is usually the major target for neoplastic transformation. So this particular virus can be transmitted, one, via sexual intercourse, especially when you're transferring infected T cells, also via blood products and even breastfeeding. The same mechanisms in which HIV can be transmitted, yeah, but remove breastfeeding because majorly there's a way they can do some medication to prevent transmission via breastfeeding, but at some point it can actually transfer HIV. So in contrast to several murine retroviruses, it doesn't have an oncogen. Remember, many viruses have oncogens, but this one doesn't have an oncogen, okay? And no consistent pattern of proviral integration next to a proto-oncogen has been discovered. So we have the HTLV1 genome contains the GAG, the POL, and the TAX gene. Remember also HIV, the retrovirus usually contains the GAG, the POL, and the ENV gene. But in addition to the three, we also have the TAX gene that is usually contained by this particular virus. Remember, the POL gene majorly calls for the enzymes, which is polymerase, integrase, and protease, envelope, and glycoprotein. But we have another one called the TAX gene, which is very important for the pathogenesis of this particular virus in terms of oncogenesis. So several aspects of HTLV1 transforming activity are attributable to the TAX gene, the TAX, okay? The protein product of the TAX gene. So remember that a gene is usually abbreviated in italics, but the protein is usually not in italics. That is one of the major things you need to know about oncology. So we have multiple HTLV1 encoded regulatory proteins, including TAX and HTLV1 basic leucine zipper factor HBZ. So remember, apart from the TAX gene, you also have the HBZ, and the HBZ and the TAX are usually important in oncogenesis. So they usually play a very important role in viral persistence and latency. And it is this viral persistence and latency that will actually lead to proliferation of different cells because it has been put under latency. And anytime we have immunosuppression, we can have a burst of this virus causing tumorigenesis. So TAX is essential for viral replication because it simulates transcription of the viral RNA from the 5' terminal repeat unit, okay? So however, TAX also alters the transcription of several host genes and interact with certain host signaling proteins. By doing so, TAX contributes to acquisition of several cancer hallmarks. So these ones, I know we have a lot of details here, but I don't want you to master them. I just want you to understand the mechanisms in which this TAX can lead to tumor genesis. Number one, this TAX can lead to increased survival and growth of infected cell, okay? And this one, number one, is due to interaction with the PI3 kinase, okay? And thereby stimulate downstream signaling cascade, which you will recall promotes both cell survival and metabolic alterations that enhance growth. So whenever you activate the PI3 kinase, you will have promotion of survival and metabolic alterations, and these ones will actually enhance cell growth, okay? So TAX also upregulates the expression of cyclin D and repress the expression of multiple CDK inhibitors. So TAX gene number one, it will upregulate the expression of cyclin D. Remember, cyclin D, a cyclin will usually complex with a cyclin-dependent kinase, and this one will actually be very important in initiation of replication of DNA, okay? But importantly, this TAX will go and repress multiple CDKIs, which are the CDK inhibitors, the cyclin-dependent kinase inhibitors. So by doing so, you'll find that whenever a cell has a malfunction in DNA, it won't be killed because the CDKIs, which are very important in counteracting the actions of cyclin D, have been repressed or downregulated. So these changes actually will lead to cell progression, and this will later on predispose a patient to cancer genesis. Then again, we also have TAX can activate transcription of nuclear factor, kappa B, which from our Kaposi sarcoma lecture, I think we mentioned quite a bit about it. It usually promotes cell survival of many cell types. Number one, by inhibiting apoptosis. And also it also leads to survival of lymphocytes, okay? So we have increased genomic instability. That's the other mechanism in which TAX will lead to tumor genesis. Remember, when your genome is unstable, we can actually have tumor genesis, especially if the DNA repair mechanisms have also been hindered, okay? So this one will lead to tumor genesis. So TAX can also interfere with the DNA repair mechanisms. And this one will actually lead to genesis of tumor because the cell cannot be put under control. So about the HBZ gene, I won't talk about it because it is sort of like detailed so much. That's why I have attached a link here that you can use so that you can obtain a lot of information about it. So for further studies about the HBZ gene, you can use this article here. So infection by the HDLV1 causes expansion of non-malignant polyclonal cell population, okay? And I think we have talked about how TAX will help in cell proliferation, so I won't repeat that. So the multiplying T cells are at the risk of increased mutations and genomic instability due to the effects of TAX. I think I've talked about that one, we won't repeat. And possibly other viral factors as well. This instability allows accumulation of oncogenic mutations. Remember, when you have genomic instability, number one, we can't repair the genome quite well, especially the DNA. So because of this, we'll have a lot of accumulation of oncogenic mutations. And later on, this one can predispose the patient to monoclonal neoplastic T cell population, which will emerge, okay? So the most common driver mutations identified thus far are predicted to enhance T cell receptor signaling and stimulate the nuclear factor kappa B. I think I've talked about the nuclear factor kappa B, how it usually inhibits apoptosis, and that is will lead to increased cell survival. But remember, these mutations majorly are driven towards the T cell receptor signaling because this virus has a tropism for the CD4 positive cells, which is a T cell. So this is just a diagram to demonstrate how how these lymphomas and leukemias can present in a patient. Yeah, so you can look at this patient here. And here we have microscopic views, how this one can be seen. We have this microscopic view, we also have this other one. Remember, these are lymphocytes. And importantly, when you look at the lymphocytes, it is not normal, it's kind of like abnormal. So you can see we have leukemic growth in this particular slide. Okay. So the second one we'll talk about is the human papilloma virus. And the human papilloma virus, first of all, it's a DNA virus, that one we know. Okay. So it usually contains different types. So we have type 1, type 2, type 4, and type 7. They usually cause benign squamous papillomas in humans, which are basically called warts. Then we also have genital warts, okay? And these genital warts, basically, they usually produced especially by type 6 and type 11, okay? That is very, very high yield for your exam. And remember, these are the low-risk HPVs. They're not high-risk HPVs. So we have two common high-risk HPVs that are usually recorded in most cervical cancer cases. And these ones are type 16 and type 18, okay? They can cause several cancers, but majorly in women, we normally see the cervical cancers, though they can also cause anogenital cancers. That is very high yield, okay? And again, in addition, at least 20% of oropharyngeal cancers, particularly those arising in tonsils, have been associated with high-risk HPVs, okay? So I want us to talk about how this HPV can really lead to tumor growth in a patient. So I want us to focus on two early proteins that are usually produced by this virus. We have E6 and E7. So this E6 and E7, they both have different mechanisms of action in terms of leading to oncogenesis. So I want us to start with E6. So we want to talk about the oncogenesis of E6. That's very, very high yield, okay? So the E6 proteins bind to and mediates the degradation of P53. So remember, E6 will act on the P53, while E7 will act on the RB gene. That's very, very high yield. So E6 will bind to the P53, and this will lead to the degradation of P53, okay? And again, it will stimulate the expression of TAT. That is very important in telomerase. Okay, it's a catalytic subunit in the telomerase. And remember, telomerase enzyme is very important in multiplication of cells. So remember, number one, if we degrade the p53 is the guardian of the genome. It usually has a very important role in ensuring genetic stability. Number one, by inducing apoptosis if a cell is beyond repair. Number two, it can lead to senescence and quiescence. So whenever you degrade p53 again, you're trying to enter the cell in terms of preventing abnormal multiplication by hindering apoptosis and many other factors I just mentioned. Okay. So telomerase enzyme usually is very important because it enables the cell to actually multiply. So whenever you stimulate that, and this is the catalytic subunit of the telomerase enzyme, it means that this will lead to immortalization of the cells. They'll keep on multiplying because of the catalytic subunit being upregulated, which is the TAT. Okay. So E6 from high-risk HPVs, they usually have a high affinity for p53 compared to the low-risk types, which usually have a very low affinity for the p53. So that's the oncogenesis of the E6. What about for the E7? So the E7 protein has effects that complement those of E6, all of which are centered on speeding cells through the G1S cell cycle checkpoint. I think we had talked about the RB gene, and we said the RB gene majorly acts on the G1S checkpoint. Okay, so one way in which the RB gene can actually regulate this G1S checkpoint is via the E2 transcription factors. Let me repeat, just a quick recap of whatever we have talked about. Okay, so we said the RB gene is very important because number one, it can be activated by removing the phosphates. In other terms, we have hypophosphorylated RB gene and hyperphosphorylated RB gene. Hypophosphorylated RB gene is usually active while hyperphosphorylated is usually inactive. Okay. Then we talked about the cyclin E. We said the cyclin E usually complexes with the cyclin-dependent kinase to form a complex which is very important for multiplication and induction of nuclear transcription. So we talked about also E2F under RB gene, and we said that the E2F is very important in transcription of cyclin E. I want you to get that point clearly. E2F is important for the transcription of cyclin E, and cyclin E will complex the cyclin-dependent kinase to form a complex which will activate initiation of DNA replication. So it means that whenever you interfere with E2 transcription factors, you also interfere with the replication. Okay, so that's how the RB gene usually works. Okay, so it binds to the RB gene. So remember, when this E7 binds to the RB gene, it will kind of like separate the RB gene from the E2F transcription factor. So when it separates the RB gene from the EF transcription factors, it means that the RB gene cannot suppress the E2 transcription factor. So the E2 transcription factor will lead to the formation of cyclin E. Then cyclin E will lead to the development of the complex cyclin E CDK complex, which will now enact the initiation of DNA replication. So that's how this E7 usually works. Okay, so because of this will lead to continuous cell proliferation because the E2 factor is not in any way inhibited by the RB due to the separation enacted by the E7 early protein. Okay, so whatever we have just mentioned here is that the high risk one has a high affinity for RB gene, while the low risk one doesn't have a high affinity for the RB gene. I think we have mentioned those also in E6, so I won't repeat that. Okay. Okay. So integration into the host cell genome also contributes to oncogenic potential. Remember, HPV is a DNA virus, so it can integrate into the genome. And whenever it integrates into the host genome, that's another way in which it can contribute to oncogenic potential of the virus. Okay. So integration interrupts a negative regulatory region in the viral DNA, resulting in overexpression of E6 and E7. So remember, this oncogenic, this integration is just important for the overexpression of E6. So whenever you are asked what is the importance of integration of HPV into the host cell genome, the answer is to lead to overexpression of E6 and E7. This is a diagram of a cervical cancer. You can appreciate it. This is a diagram of the cervical cancer. And this one will just show different types of cancers that can be caused by this particular virus. We have the cervical cancer, which will come from the cervical precancer. Then we talked about the oropharyngeal cancer, which is rare though, and we talked about the anogenital cancers. For example, we have the anal cancer, genital, so the vulvar and the vaginal cancers for female, and the penile cancer for men. Okay, so let us talk about the Epstein-Barr virus. So the Epstein-Barr virus It's also a member of the herpes virus family, okay? And it is the first virus to be linked to the causative agent of tubercular lymphoma. So EBV has been detected within the cell of a surprising diverse list of tumors, including nasopharyngeal carcinomas and different types of T-cell carcinomas, and even some gastric carcinomas. So remember, EBV has been associated with nasopharyngeal carcinomas, bucket lymphomas, T-cell lymphomas, NK cell lymphomas, and gastric carcinomas. Okay, so let us go to the mechanism in which this one is caused. So EBV usually has a complement receptor called the CD21, otherwise called the CR2. So remember, for these viruses to cause tumor growth, they need to bind to a receptor before they enter the host cell. So EBV will use the complement receptor CD21 to attach and to infect the B cells. So it will attach and to infect the B cells. Okay. So in vitro, such infection leads to polyclonal B proliferation and generation of immortal B cells. So we know that in vitro is outside the body. So whenever you're doing an experiment and you try infecting maybe this virus to a B cell, it relates to a polyclonal B cell proliferation and generation of immortal lymphoblast cell lines. Immortal in that they'll continue multiplying and they won't be killed quite easily due to Darwinian selection. So one EBV encoded gene is usually the latent membrane protein 1. This is very important. This is one of the EBV encoded gene, and it usually acts as an oncogene. Okay, but remember we said that the HTLV1 doesn't have an oncogene. Okay, so this LMP1, which will act as an oncogene, as proven by its ability to induce B-cell lymphomas in transgenic mice. So whenever you take this LMP1 and you kind of like how can I put it? You kind of like inject it into a B cell, it will lead to formation of B cell lymphomas in transgenic mice, okay? And this LMP1 also is very important in promotion of B cell proliferation. And this one will mimic the effects of a key surface receptor such as the CD40. I think we talked about the CD40 ligand, especially in the immunology classes. We have the CD40 and we have the CD40 ligand, okay? So the CD40 ligand is usually expressed in the T cells, okay? And this CD40 ligand will bind to the CD40 in the B cell. And this one is basically important in B cell colonial expansion, okay? So this one will form another means of signaling in the entire cascade of B cell proliferation. So by contrast, LMP1 is constitutively active and stimulates signaling through the nuclear factor kappa B. We also have the JAK-STAT pathways, both of which promote B cell survival. Remember, this and nuclear factor kappa beta is very important. That's why in every tumor we have talked about it, okay? So number one, you said that it will inhibit apoptosis, and this one will lead to increased survival of the cells. We also have the JAK and STAT pathways, and these are usually related to the tyrosine kinase family of cytokine receptors, okay? So another EBV-encoded protein is called the EBNA2, and this EBNA2 usually activates, this is very important, it usually activates cyclin D and the SRC family of proto-oncogenes. Remember, cyclin D is very important for the multiplication of the proliferation of a cell. And I think we have talked about it. Whenever you see a cyclin will actually form a complex with a cyclin-dependent kinase to form a complex that can activate initiation of replication of a cell. So that's why this particular encoded protein will actually activate cyclin D. And the importance of activating cyclin D is that this one will lead to increased proliferation of B cells by increasing multiplication. Okay. In addition, EBV genome contains a viral cytokine, and this viral cytokine is usually called the V interleukin 10. So this VIL10 is usually very important in terms of preventing destruction of these cells that have been infected, okay? So it will prevent the macrophages and monocytes from activating T cells that will kill the virally infected cell. So remember, it is a virally infected cell that later on will develop into cancers, right? Yeah, so that's the importance of this. This V interleukin 10 will actually prevent macrophages and monocytes from activating T cells. And because of this, the infected cells will be spared, which will form one of the bases of survival, and this later on will lead to expansion and development of lymphomas. So a small number of EBV-infected cells can regulate expression of immunogenic viral proteins, LMP1, and this is not high yield, so I won't say so much about it. What I want to talk about again is this thing called malaria. So we have in some regions where bucket lymphoma is endemic, and this is because of concomitant infections with malaria. So malaria will impair the immune system, making the person to be immunosuppressed. So whenever you're immunosuppressed, this virus now will now be amplified to lead to the development of bucket lymphoma. Okay, this one will lead to sustained B cell proliferation. Eventually, cytotoxic T cells eliminate most of the B cells, because remember the immune surveillance is always there to also kill the cancer cells. One example is the tumor necrosis factor. It was called tumor necrosis factor, tumor necrosis. It was believed that it was killing tumors by necrosis. Okay? So we have the immune surveillance, which will kill most of the infected B cells, but some of them will survive. A small number of them will survive. Not all of them will be killed. The same thing can be applied to maybe a war. When people are fighting, you can kill literally everyone, but you'll find that 2 or 3 people will survive. The same thing can actually be, can also be related here. So virtually all endemic and sporadic cases of bucket lymphoma exhibit T814. So this is basically a translocation from position 8 of the chromosome to 14, okay? And now this translocation called the 814 will dysregulate functioning of the MYC. And remember, MYC gene was very important together with the likes of Jun and even FOS, okay? In contrast to bucket lymphoma, tumors in immunocompressed patients usually lack the MYC translocation. So remember, this bucket lymphoma can even arise in patients who are not immunocompromised, okay? But majorly, when you look at the tumors arising from those who are immunocompromised, they usually lack the MYC translocation. But bucket lymphoma must have a MYC translocation, okay? Yeah. So these potentially lethal proliferations can be subdued if the functioning of T cells is restored. So remember, if these patients are immunocompromised, it means that the functioning of the T cells kind of is downregulated, right? So for us to prevent these lethal proliferations of these cells, these B cells, to lead to lymphoma formation, then it means that we actually need to activate or restore the functioning of the T cells. So in case the functioning of the T cells have been restored, it means that the potentially lethal proliferations will actually be subdued, okay? But remember, 100% of nasopharyngeal carcinomas contain EBV. But remember, not all bucket lymphoma are caused by EBV, but all cases of nasopharyngeal carcinomas contain EBV. Now, this one is showing that EBV is essential for the genesis of this type of tumors. I repeat, not all bucket lymphomas are caused by EBV, but almost 100% of nasopharyngeal carcinomas contain EBV. And now this one will show that EBV is essential for the genesis of this type of tumors. Again, another difference is that unlike bucket lymphoma, LPMP1 is expressed in nasopharyngeal carcinoma cells, and as in B cells, activates the N nuclear factor kappa B pathway. Remember, whenever you activate nuclear factor kappa B, apoptosis will be prevented, and this will lead to the survival of the cells. Okay, this pathway can dysregulate apoptosis pathways and upregulate expression of factors such as the vascular endothelial growth factor. Let me explain this point. Remember, we have the guardian of the genome is called the P53, and the P53 is usually very important also in anti-angiogenesis. The P53 usually helps in the production of thrombospondin 1, and thrombospondin 1 is very important in acting as an anti-angiogenic substance, okay? But whenever we have tumor genesis, the P53 will be downregulated. So whenever you downregulate the P53, it means that thrombospondin 1 cannot be formed, and it means that vascular endothelial growth factor cannot be antagonized by thrombospondin 1. So this is what will lead to vascular endothelial growth factor overexpression, and this will lead to neovascularization to feed the new tumor. Because remember, for a tumor to grow past 1 or 2 millimeters, it normally requires blood supply and nutrients. Okay. So virtually, when you look at some cancer therapy cases, whenever you want to treat a patient of cancer, you can target the neovascularization, okay, by administering drugs that can counteract neovascularization. This is a case of a bucket lymphoma. You won't love the way it is presented. So yeah. Then write down as a case of an nasopharyngeal carcinomas. Yeah. I think you can appreciate this. This is the right nostril. You can even see the right suction tube orifice, the suction tube opening. So this basically is a nasopharyngeal, from the nasopharynx, naso from the nose to the pharynx. So let us finish the hepatitis B and C very fast. So hepatitis B and C, they normally cause 70 to 85% of hepatocellular carcinomas, okay? So the HPV and HCV genomes do not encode viral oncoproteins. And although the HPV DNA is integrated within the human genome, there's no consistent pattern of integration in the liver cells. That is not IEL, so I won't talk about it. Remember, I'm just talking about the IEL points. The oncogenic effects of HPV and HCV are multifactorial, okay? But the dominant effect seem to be immunologically mediated by chronic inflammation with hepatocyte death. Remember, from your microbiology classes, we say that hepatitis B and C, they normally cause their pathogenesis majorly by chronic inflammation. So whenever we have that chronic inflammation, we can lead to death of hepatocytes, right? And remember that whenever we have a damage to the liver is usually in the GO phase, but whenever it has been stimulated, it can lead to overproliferation. You know that the liver can actually proliferate again, okay? So whenever we kill the hepatocytes, some of them will try to arise again. Some of them will try to be formed again. But in the process of being formed, they can be formed in an haphazard manner, okay? So this now will lead to, okay, let me just repeat. Chronic inflammation with hepatocyte death will lead to regeneration and genomic damage. That's what I was explaining, that whenever we have that rapid regeneration, we can have genomic damage because the cell is multiplying so fast so that even the p53 and the repeating functioning can be a bit impaired, okay? So although the immune system generally is thought to be protective, recent work has demonstrated that the setting of unresolved chronic inflammation that can occur in viral hepatitis, okay, the immune system may be maladaptive and promote tumorigenesis. And I think we have talked about how it can promote tumorigenesis, basically by increased regeneration, which can lead to genomic damage. And whenever we damage the genome and the cell is still multiplying, we can have tumor growth, okay? As with any case of hepatocellular injury, chronic viral infection leads to compensatory proliferation of hepatocytes. I think we've talked about that one. This regenerative process is aided and abated by a plethora of growth factors, cytokines, and chemokines, and other bioactive substances produced by activated cells that promote the cellular survival, thus leading to tissue remodeling and angiogenesis. So remember, regeneration of a tissue is usually directed by the number one growth factors. It can also be by cytokines, things like chemokines, and even bioactive substances, okay? So remember, whenever I talk about a bioactive substance, these are substances that have the propensity of even causing reactive oxygen species. When you're bioactive, you can cause ROS. And know that ROS are usually genotoxic and even mutagenic. ROS can lead to damage of the genome, and they can even lead to transformation of a cell, okay? So a key molecular step seems to be activation of the nuclear factor kappa B. We have talked about this one repeatedly. But whenever we have nuclear factor kappa B, this one will inhibit apoptosis, okay? So whenever we activate this nuclear factor kappa beta pathway in hepatocytes caused by mediators derived from the activated immune cells, for example, things like cytokines, chemokines, and even the growth factors, okay? So activation of this pathway will block apoptosis, okay? It will block apoptosis, allowing the dividing hepatocytes to incur genotoxic stress, okay? So the hepatocytes is actually doing what? Is dividing. But in the act of dividing, even cells that have genomic damage are being multiplied rapidly, okay? So we know that in common sense, your cells usually have different ways of repairing DNA, right? But because you have the NF kappa beta pathway, which will block apoptosis, now these cells, because they have been damaged by genotoxic stress, they can't be repaired, and again, they can't be taken to the quiescence phase. The only thing that can happen is they can be killed by apoptosis. But now remember that the process of killing by apoptosis has also been prevented by the NFKB, the nuclear factor kappa B. So this one will actually lead to accumulation of mutations, and later on this one will predispose the patient to cancer. I want to talk about a very important gene called the HBX gene, and this one is found in many hepatocellular carcinomas. And an experiment has been done where the HBX gene was engineered to a mice, okay? And whenever you engineer it to a mice, the mice will develop hepatocellular cancer. So this one will show that this HBX gene is very important in hepatocellular cancer genesis, okay? So how does this HBX protein help in the entire thing of tumor growth? So number one, it will lead to interference with the p53 function. And whenever you interfere with the p53 function, which is the guardian of the genomic stability will be interfered with, okay? So in addition, also, we have the viral integration. Remember, whenever we have viral integration into the genome of a cell, this one can actually lead to increased chances of tumor growth. So in addition, viral integration can cause secondary rearrangements of chromosomes, including multiple deletions, and this one may actually harbor tumor suppressor genes. So that is basically how these hepatitis B and hepatitis C will lead to tumor growth. Remember, HBX gene is very important in tumor growth, and also the ROS will lead to destruction of the genome, so it is genotoxic and again mutagenic. And we also have another one called the nuclear factor kappa beta pathway, which will prevent apoptosis. And whenever you prevent apoptosis, the abnormal cell will keep on multiplying, and this one is what will lead to development of tumor. So those are the most important points that you need to know about hepatitis B and hepatitis C. They have this sort of like the same mechanism of causing cancer. So here I was just trying to demonstrate how these things happen. So I think I've just mentioned them. So I won't repeat this diagram. It's a very easy one to understand. And here we have an image of an hepatocellular carcinoma, which is kind of like appreciative. So that's all that I had for viral oncogenes. The series for Kaposi sarcoma, I had done it before. So I'm sorry I didn't merge it with this one. So maybe it can be a little bit inconveniencing, but I beg your pardon. Yeah. So thank you very much for your time.